Today is our 14th webinar, and I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, a group of companies that are dedicated to change healthcare through functional medicine, personalized medicine, and so on. We have a lot of followers, so a lot of you already know each other and you can recognize each other's names, and that's really the purpose also with these webinars, that you don't feel alone with all this knowledge and interest and so on, but that we can meet in this community to learn more, and you can exchange in the chat as well. You can also ask questions in the chat, because when we finish the talk with Sebastian, you can put Q&As, you can put questions, and then we'll get the answers in the end. So right answers, I'll put them down and take them at the end of Sebastian's talk. So remember, you can still make it for our September DNA Life training if you're interested. We will, of course, send links out so that you can sign up for this. That was based on the Sherwood's webinar that we did 2 weeks ago. And I will have another webinar, of course, for you in another 2 weeks' time. We have a planning meeting tonight. It's going to be really exciting. So, but of course, we will send out invites as soon as possible. So, but anyway, the star of the show today is Dr. Sebastian Brandhorst, PhD Research Assistant Professor of Gerontology at Southern California University. And his specialty is fasting mimicking diet. And myself and my husband and partner in crime in Nordic, together with my sister and her boyfriend, we went and we were actually invited to go and visit the research lab. And it was Sebastian who opened the doors to us, and he showed us the whole research unit and so on. And it was very, very exciting. And Sebastian has a way of explaining some of these mechanisms and talking about fasting mimicking diet in a way that's it just makes it very simple and easy to understand. And I was like, why don't I know this guy already? But I think he's hiding a little bit in the research lab. So I'm maybe a little bit unfair pulling him out here, but I just really want to share Sebastian with all of you lovely people out there that I enjoy working with. So yeah, Sebastian, it's your go.
Great. Well, first of all, thanks for having me. It's always a pleasure to share the work that we're doing with everyone. And obviously, I do give quite a bit of talks, but I've been very limited to the US. Valter Longo, my boss, he's usually the one that is all over the world, and I get sort of the breadcrumbs sprinkled out over the US. So I'm gonna start sharing my screen, if that's okay, and get into it. Okay, so let's see. So the title of the talk that we decided on was the Fasting Mimicking Diet, Recent Scientific Research and Updates. And you see my conflict of interest is here. My boss, Valter Longo, has also founded the Create Cures Foundation, and I am also working as the scientific director for that. So that's straight ahead. You have that disclosed. But what I really wanted to do today is sort of outline how we explored and exploited potentially nutrient sensing pathways to modulate health and longevity. I'm going to give a little bit of like the outlines that were behind the development of the FMD, so the fasting mimicking diet, and the different effects in disease models. So that's largely preclinical. A lot of the work that we do, given that we are a research lab, is still sort of the preclinical research arm in mouse models, but we're also involved actively in probably over 20 clinical trials worldwide now, and I will comment on some of those towards the end of the talk. And so this is designed to give you an overview of the areas that we're actively working in, because that sort of also directly outlines the directions that we're moving forward with for now. That being said, there's a lot of turnaround in the lab, and you know, directions can change from day to day. That is also part of Valter's personality. The reason why I put this little cartoon in here is a lot of the work that we work on are really based on using fasting as an intervention. My PhD thesis was based on the use of periodic fasting to improve the efficacy of chemotherapy in cancer treatment. So that's sort of 10 years ago where we were with this, and this really was the basis and the foundation for developing the fasting mimicking diet. So I'll start walking you through it. I'm a research assistant professor at the School of Gerontology. So obviously, I think all of you are familiar with the term of gerontology, but it's really the study of old age, the process of aging, and the problems that arise with it. And so it is, at the school here, we're both tackling the social concepts of aging. The focus of our study group is really more on biological concepts of aging, and sort of in the cartoon here, you can sort of see the progression as we go from young adulthood into an elderly person, and sort of the drivers, the motivators, and what makes us essentially go right through these periods. Aging obviously is not just a phenomenon that occurs at an older age, but a lot of the processes start at a younger age and sort of grow and expand over time. And that is really why aging is the catalyst for all chronic diseases that we see nowadays, right? So if you just look at the top three here, neurodegenerative diseases, cardiovascular, cancer. So these are really the sort of main killers in all of the developed countries. And if you really understand the concept of these diseases, for example, cancer requires the accumulation of mutations over time. So time is the main driver for these diseases. And so aging itself really is a catalyst for all of these diseases. That also means that if we understand how we can modulate aging, meaning the pace and the quality, this is a powerful intervention to increase health span. So the main idea of the research that we're doing is not so much focused on extending longevity by all means. We're not trying to make people live to 300 and everybody is miserable, but the concept that we're pursuing is to make people live healthy in place, right, where they with their maximum sort of health that they can reach to get to an old age. And so as a comparison here, you can sort of see two different life curves, let's call it this way. The bottom one really is here sort of standard US, right? You're overweight, stressed, fatigued in your 20s, and as you age, this all accumulates and leads to essentially the development of many of the aging-related diseases, arthritis, diabetes, high blood pressure, and ultimately osteoporosis, dementia, cancer, and so on. But at the same time, we all know that you can maintain active well into an old age. And there's just prime examples for this with, you have a balanced lifestyle, you're energetic, active. And so that means that even well into your 60s, you could be fit and active. And then we have this sort of phenomenon, which we call in aging research a squared curve, right? So you maintain this optimal health for a long time, and then you have a fairly dramatic decrease in this quality of life. That itself is not necessarily bad. It really means that you're getting to the point where your body starts to become frail and fail, but the period that you're living with multiple aging-related diseases is short, right? So you're not having, like in this lower part here, where you have a longer period of your life spent in average or very poor health. So technically, this squaring of a curve is a good phenomenon. The work that we do is focused on using diet and nutritional approaches to sort of traverse back from one curve to the other by developing healthy aging programs. And again, our focus is diet and nutrition, but everybody that's sort of familiar with the concept of the blue zones, meaning these hotspots all over the world where we see extended longevity compared to the surrounding areas. If you ever looked into this research, there are obviously very important concepts to be taken away from that are not just diet and nutrition, but it's a lot of focus on rest and relaxation, sort of the social connection, intellect, exercise obviously to a degree plays a role, spirituality. So it's really more like a holistic approach. We just happen to focus on diet and nutrition because that is something that can be easily modified. A lot of these concepts of social connection also is a lot harder to modify, and quite frankly, not all of them are biological approaches. So dietary approaches to improve health and longevity are nothing new. Caloric restriction has been used in mouse research for nearly a century now. And first it was sort of developed or tested in mouse models with cancer, but since then has been shown to extend longevity in many of the rodent models. There's a little bit ongoing conflicting data whether or not it increases overall longevity in monkeys, but there's a lot of great data really showing that the effects on health are generally preserved when looking at caloric restriction. The definition of these diets are here. I'm not going to go through all of them because I really want to spend more time at the bottom here. But obviously, time restricted feeding is something that has been come up in the last sort of maybe decade. A lot of focus on aligning your food intake with the light-dark cycle. Also has shown some good results against diabetes. It helps with weight loss, right? Then some more I guess human approaches that are very popular right now: intermittent periodic fasting. So two different approaches that just modulate when and how you eat, and it's essentially a fasting day followed by feeding days for the intermittent fasting, or sort of a periodic fast, which could be a version of the 5-2 diet, for example. And again, here we see an increase in median life expectancy in mice, as well as effects on health. And then at the bottom here, this is what we developed at USC, and I will comment a little bit later on it, what we do with it. But this fasting mimicking diet is really a sort of a meal plan, right? In humans, it's a packaged diet. For mice, we use now actually the same sort of human product. We grind it up and feed it at appropriate calories. It's essentially a reduced caloric intake for 5 days, and we're also modifying the macronutrient composition. So it's generally a low protein, medium to low amount of carbohydrates, and then it's a fat-rich diet, but it's not quite a ketogenic diet. And we've shown protection from cancer and diabetes, improved risk factors for age-associated diseases, and so on. And I will comment on the preclinical research in the sort of upcoming slides, and then I'll show you some of the results that we have in humans. So the important thing is that unlike many drugs, the modulation of food intake sort of has this really, really broad approach, right? So we're starting in the middle here, feeding fasting cycles, and it starts to modulate a lot of these hormonal regulations, right? Insulin growth factor 1, but also other energy sensing molecules in the body, and release of fatty acids and so on. And that itself then has a systemic effect on metabolic homeostasis, overall function, tissue repair, and resilience. So this is really one of the sort of powerful approaches of why dietary approaches are so good in really having a really broad effect. A lot of times people would ask me, like, why do you think the one diet that you work with affects everything, right? I talk about diabetes, cancer, longevity, rejuvenation, and the answer is right here. It's literally you're targeting this circle right in the middle, and you modify not just one mediator, but all of those with the diet. So that is important to keep in mind. Obviously, as humans, we have evolved, and we're actually quite good at dealing with fasting. So we're not reinventing the wheel here, but we're using evolution that is integral for us. So throughout our evolution, we're not exposed to food all the time, and so fasting is actually, we undergo cyclic fasting relatively often. 99% of the time we're used to fasting. That is also displayed sort of by the transition that fasting is beneficial, as shown in many of the religions. So you could think about Lent or Ramadan fasting. So there was inherently something about fasting and the withholding of food that has a concept in religion that it's good for you. However, we have replaced fasting in the last 50 years, and instead now food is available for 24/7, meaning you can eat whenever and however you want. And there are concepts of it that make it really bad. From it's like when you look at shift workers, that their food intake is sort of misaligned with when their hormones are actually working optimal to digest. So there's issues here, and that's really been leading to this increase in the obesity pandemic. up on top here, you can sort of see as the colors change this is for the United States, and so a little bit updated. It's only going until 2013, but you can see sort of the increase in obesity in the U.S. adults, right? And this is particularly prominent sort of like in the southern states here, Louisiana, Alabama, Mississippi, Texas, and that is obviously associated with a higher intake of sugar sweetened beverages, fried food, and so on. And then you can see that in hand with this obesity pandemic, there's an increase in particular cardiovascular diseases and cancer predicted for 2020. So this publication is from 2004. So again, it's old, but it's still holding true. We're actually seeing a dramatic increase in obesity still in the United States, up to the fact that actually for the first time, I think last year, it was published that the life expectancy in the United States is decreasing for the first time because just the obesity pandemic has taken a toll. And a lot of the diseases are obviously driven by obesity as well, right? So cardiovascular disease, obviously obesity is a major driver of that. So here's just a quick comparison. So today's food, at least in the United States, I'm not sure if this is how true this is for Denmark right now, but I'm assuming it's comparable. So you know, we have high. First of all, we have high caloric intake. So a lot of times people don't even know what they're eating anymore with regards to their calories that they're consuming. But then also all of these diets, we have a high intake in protein, sugars, and carbohydrates, and all of those activate a intracellular cascade that is associated with unhealthy aging. I will comment back on that in the next slide. Whereas fasting or the fasting mimicking diet have actually low amounts of sugars, low amounts of protein, which in turn deactivates these pathways and optimizes aging. And so this is just an idea, and it gets a little bit more complicated here, but it's actually quite simple to understand. So we sort of go through evolution from yeast, worms, flies, mammals, and as we go, these nutrient sensing pathways that are depicted here get a little bit more complicated. So in yeast, they essentially sense directly glucose and amino acids in their environment. In mammals, this is usually done through hormonal signaling. So particularly growth hormone is in mammals is released in response to protein uptake. But all of these here, you can see sort of the same patterns indicating that these are orthologs, right? So we see them throughout evolution. They have the same function and we find them. And all they do is that when nutrients are abundant, these pathways get activated and that leads to cellular growth. That also means that in this particular case, cellular growth equals cellular aging, right? So if you consider something like the Hayflick limit, which means that a single cell has sort of a limited amount of replications that it can do, that means that the cell is efficiently or essentially aging. What this also shows is that by restricting the amount of glucose, amino acids that is around, or reducing these hormonal signaling, this in turn deactivates these pathways, and that leads to an induction of protective metabolites and metabolic activities that are associated with an increase in lifespan. So you essentially you can imagine these pathways like a switch, and meaning when this food is abundant, this whole pathway promotes cellular growth. When food is not abundant, it's been deactivated, and it's been just simply put into a more resilient cell state. So under these conditions, under fasted conditions, cells can deal with a lot higher sort of insults and toxic assaults. And that is actually what that was sort of the concept that we played with for the work that I did 10 years ago, which was that you can use the modulation of these pathways to increase the resilience of cells against toxic insults. So the work that I did was you can give animals very high doses of chemotherapy to a point that it's toxic to them. And if you don't fast mice, they unfortunately will die from that attack. If you fast animals just for about 60 hours prior to giving them the same dose that killed all the fat mice, nearly all of these mice survive. So it's a really interesting approach to sort of play with the organismal resilience. There's another concept to this, and this is, I think it's a little bit important for later. Obviously, fasting is done periodically. That also means that there is going to be a time point where you stop the fasting and you are refeeding. So you're turning these pathways back on. And we have shown repeatedly now that this leads to an activation, particularly of stem cells. Some of them are residing in the hematopoietic niche. Others can be found, for example, in the liver. So starvation leads to an increase in lifespan. This might be initially counterintuitive, but you can see this here. So bacteria, when you starve them, outlive their fat counterparts. The same is true for yeast, and we see this also in C. elegans. So this round worm here, the red line is essentially you starve them of bacteria, and they all outlive their control fed animals. So inherently, starving animals for a restricted period of time is a good thing, and we can mimic these periods of fasting by inducing mutations in these nutrient sensing pathways, right? So you can see here, this is a colony of yeast cells. They're smaller. They have a mutation within one of these nutrient sensing pathways. The same is true for the fruit fly here and mice. The interesting thing is that yes, they're smaller. Again, we're inducing deactivating mutations in these nutrient sensing pathways, so therefore growth is stunned. But all of these shorter animals here live longer and have less of the aging related diseases. This mouse here actually holds the world record sort of for the lifespan extension. The mouse lives about 60% longer, and the majority of these mice actually don't die of aging related diseases. So it's a very interesting concept. And there is an equivalent to these mutations actually found in humans. So this is my boss here, Valter. You can see it's an old picture, but there is together with a collaborator in Ecuador, there's a population of this is called Laurent syndrome. So they have a mutation in the growth hormone receptor. So it's essentially going back here, growth hormone is released by the pituitary gland. And then through the growth hormone receptor largely in the liver, this leads to the production of insulin-like growth factor 1. So these subjects have very high level of growth hormone, but because they're lacking the growth hormone receptor, this signaling cascade here never gets turned on, which leads to stunned growth. So you can see this here. It's a proportional form of dwarfism. The really interesting thing is when you compare these subjects to their relatives in Ecuador, these growth hormone receptor deficient people, compared to their relatives, there's no incidence of cancer and there's no incidence of diabetes, which is remarkable given the fact that a lot of them are quite overweight or even obese. So there's a relevance of these nutrient sensing pathways and the modulation of it for humans, right? Again, I'm not saying that this is the approach that we're taking, but I think it's highlighting the importance of these pathways and what it could mean for human aging. So I think the importance of doing this research is to identify what is actually feasible in humans. And a lot of these dietary approaches, a particular caloric restriction, has been tested before. There's multiple experiments that usually indicate really high dropout rates, and it's really hard to do for those people. There's some people, I think they're called the cronies, and they're doing chronic caloric restriction, and it's a very, very dedicated few people. Our approach has been sort of, we want to identify something that's actually doable by a lot of people, and so you can see here the criteria that we based this on. We wanted to have a short intervention, so people can you do it, you're done with it, and you can come back to it. So it's periodic. It's not consistently intervening with your lifestyle. It should also align with good anti-aging practices. So the idea is not just to pick up any sort of trend and play around with it, but we really want it to be based on good anti-aging practices. Valter always calls it sort of like the 5 pillars of longevity. And I think an important concept is also that it should be integrated into your social life. So I think a lot of the diets fail because people are unable to continue to do them once they sort of fall back into their normal rhythm. And so the diet that we developed is low in protein. The main reason here is, again, we don't want to activate the growth hormone IGF-1 axis. It's low in carbohydrates. The FMD was initially designed to to treat cancer, right? So we had a lot of great data using fasting in cancers, but the oncologist and the treating doctors essentially refused to have their patients for extended period of time just do water only fasting. There was a little bit of fear of malnutrition just coming from the water only fasting. So in this case here, we have a low carbohydrate diet. That is because most cancers have a high dependency on glucose. It's vegetable based, and it's obviously calories have to come from something. In this case, we're using a relatively high amounts of polyunsaturated fatty acids. Okay. Sorry, there we go. So up to this point, this is sort of an overview of the publications and the work that we've done. I think we take great pride in having our research peer reviewed, and I think, as you can see, sort of from the caliber of the publication and their impact factor, I think we're usually doing really well. So there's Cell Metabolism, Cancer Cells, the reports, the Science, Translational Medicine. So overall, a lot of this we've really shown now in preclinical and clinical trials that the fasting mimicking diet is affecting against aging, and also multiple of the chronic diseases. And these are sort of like our last publications are those last three here, and I will comment a little bit on those as well. So I'm going to walk you through sort of the initial idea behind the fasting mimicking diet in mice and what we have done, and then go to the humans, right? So I think for the first step is to actually make sure that the fasting mimicking diet indeed is mimicking fasting. So this is the concept here. We're showing this is 3 days of water fasting in mice, and this is 4 days of the fasting mimicking diet in mice. These markers that we look at here, like I said, are in part measuring a response to the fast. So glucose obviously decreases rather quickly when you start fasting. IGF-1 was the insulin-like growth factor that I mentioned earlier, which is downstream of the growth hormone. So if we see a decrease here, we know we're deactivating these nutrient sensing pathways. On the same hand, we know that with fasting, ketone bodies increase, so we should see an increase in ketone bodies. And IGF-BP1 is the binding protein for IGF-1. So just by increasing the levels of IGF-BP1, we're essentially blocking and binding the availability of IGF-1. So these two need to go in hand. And you can really see when you compare it here, there's similar effects on glucose, ketone bodies, IGF-1, and IGF-BP1. So in mice, the fasting mimicking diet for 4 days, 3-day water only fast in mice. So this was sort of the first study that we published with it, and I'm summarizing probably 3 to 4 years of research in this one slide here. It's essentially a lifespan study. And what we did is we started middle-aged female mice on this fasting mimicking diet. And the way we did it was we put them on the diet for 4 days, and then they were allowed to eat whatever they wanted for the next 10 days, and then repeated these cycles. So you could sort of see here a summary of what's been going on. What we first demonstrated was sort of a multi-system regeneration. I'm giving you an example here of the liver because it's the most prominent and it's the most dramatic, but you can see this sort of the same pattern for multiple other organs. So essentially what you see here in old animals, so these are 20-month-old mice, they have been on, the FMD animals have been on the diet for 4 months at this point. So you can see that directly after the fasting mimicking diet, so after 4 days, liver weight shrinks quite dramatically, and this is largely in part because the fasting mimicking diet induces autophagy. So we see a drop here, but then the moment we're refeeding these animals, so this is a week after refeeding, the liver weight is completely restored. So meaning we're losing cells, but the moment we're starting the refeeding again, now younger or cells within the liver start to re-expand and regenerate the liver mass that was lost. You can see this here by sort of measuring the amount of proliferation that is happening in the liver. You can see This is the sort of basal level under normal feeding conditions. This is 1 day of refeeding, so we're having a dramatic increase here. This consists for about 3 to 5 days, and then after 7 days, these levels are normal again. So really important thing to take away is not just effects of the immediate fasting mimicking diet and the low calories that goes in hand with it, but it's important to note that there are benefits from refeeding. These are here on the below it. You can see the this is measuring the fat mass in these mice. In gray here, this is the subcutaneous fat, so you actually kind of want this to be there. In red, this is the visceral fat, so this is the bad fat surrounding the organs. And you can see in the fasting mimicking diet cohort, fat mass was dramatically reduced. So these animals are a lot leaner, a lot healthier. And this is not at the immediate end of these 4 days of fasting mimicking diet feeding, but this is actually after refeeding. So they maintain a very lean physique throughout their life. When we look at the number of tumors, you can see here a dramatic reduction in all sorts of tumors. It's important to note that there is no difference in the type of tumors they're getting. They're just getting less. We see a reduction in inflammatory diseases. We see rejuvenation in the immune and cognitive system. You can see here, these are white blood cell levels in these mice. These are young animals. These animals here in light gray, they're about 2 to 4 months old, and then you compare them to the old ones. Numbers here are quite dramatically reduced. However, in the animals that were maintained on the fasting mimicking diet, at least during the refeeding, these levels are comparable at 20 months to a 2 to 4 month old mouse, right? So again, indicating here that we're storing these white blood cell levels. And overall, this led to an increase in medium lifespan, not so much at older ages. And there's an important component to this, which I'm not going to comment too much on it, but there's now emerging a lot of emerging information coming to or leading to the conclusion that the nutritional requirements change with aging. So while we have a lot of benefits sort of at the middle age to young old mouse, we actually saw they struggled quite a bit with regaining their weight in older animals, particularly after this fasting mimicking diet. It took them quite a while to recover. So we actually stopped feeding them the FMD at around here. But that being said, in some of the clinical trials, we're having subjects in it that are about 70 years old. They actually respond really well to the FMD. So I'm going to shortcut through a couple of things that we're doing in the lab or have been doing in the lab. Obviously, one of the major components of our research is still focused sort of on cancer management. Showing you here results from a study that we published in Cancer Cell in 2016. And essentially, the takeaway message from this is you're having different cancer models here. This is a breast cancer treated with either cyclophosphamide in the top panel or doxorubicin in the middle panel. And then at the bottom here, you have a melanoma. And it's important to note that while the drug itself is always doing what it's supposed to do, namely reducing tumor progression, we can see that the fasting mimicking diet alone, without the combination with chemotherapy, is in some cases as effective as toxic chemotherapy in reducing this tumor progression. You can see this quite nicely here and here, and obviously also in the melanoma. But the real benefit of having the FMD is when you combine it with chemotherapy. So we see a dramatic reduction in tumor progression over time here in these mouse models for both a breast cancer as well as a melanoma model. And these are the bar graphs are just a readout at the end of the experiment. So you can see in these shaded barred graphs here, these are animals that were treated with the FMD and the drug, and compared to the drug alone or the untreated mice, you have really dramatic increase in the efficacy of the drug. We also used the FMD in a type 1 and type 2 diabetes mouse model. I'm showing you here the results from the type 1. And the way this model works is quite simple. You have animals, you give them STZ. So this is actually a diabetes-inducing drug, which by killing the insulin-producing beta cells. And so you can see you give them the drug here, and these black dots are animals that are just treated with the drug, and these are blood glucose levels. So they're essentially getting hyperglycemia, really high levels, and then at around this point, the majority of these mice actually has died off because of their high blood sugar levels. But you do the same treatment, and then you just give them, and these dotted lines here are 5 cycles of the fasting mimicking diet. And you can see that already after the first cycle, these numbers sort of decrease. After the 5th cycle, they're pretty much back to normal. And the interesting thing here is when we look at this, this is what your pancreas should look like, or at least the mouse, not yours, but the mouse one. This is following the treatment with the drug. So you can see that all these red insulin-producing beta cells are nearly gone. And these are the effects that we see when we combine STZ plus the fasting mimicking diet. So after 50 days, we essentially restored insulin-producing beta cells in the pancreas, which explains this decrease in blood glucose levels and also increases their survival. And the same effects were actually true for type 2 diabetes model. We used the fasting mimicking diet in a mouse model of multiple sclerosis without going into too much detail here. So EAE is an experimental autoimmune encephalomyelitis. So essentially, we're giving them a drug which leads to the demyelination of their axons. And you can see in black here, these are animals that were treated with just the EAE, and we're measuring sort of a clinical scoring here. And this is measured as paralysis. So 3 to 4 means that 3 is paralysis of one hind leg, 4 is 2 hind legs, 2 is sort of the there's paralysis in tail, 1 is just like the tail is tipped. So you can see that the these animals exposed to the EAE have quite dramatic symptoms of multiple sclerosis-like symptoms. We compared this to a ketogenic diet, which is the red here, which is thought to have autoimmune properties, and it's reducing the effects of the in this model here. But at the bottom, these are 2 different approaches with the FMD. One is a semi-therapeutic, so the animals were treated once 10% of the the animals were exposed to the FMD once 10% of them showed MS-like symptoms. And then in a T here is the therapeutic. So these animals were treated once each mouse developed MS-like symptoms. And you can see that overall there's a little increase and then a decrease here. But when you compare these effects here, the FMD is really powerful in reducing multiple sclerosis-like symptoms in this mouse model compared to just a ketogenic diet, or even more so, obviously, to the control fed animals. And we also used the FMD in a mouse model of inflammatory bowel disease. So this is the agent that we're giving here is DSS. So when you see it down here, you know what it is. So essentially, what you do is you give the mouse DSS in the drinking water, and over time, this leads to the development of inflammatory bowel disease in this mice, and you would sort of see a reversal. You see a short intestinal shortening, high levels of inflammation, reduction in sort of stem cells residing in the crypt here in the gut, and changes in the microbiome population that are just coming from this IBD model. When we combine it with a fasting mimicking diet, we actually see a reversal of the intestinal shortening, partial reversal of the inflammation. We see an induction in stem cells residing in the crypt, and we're seeing a change in the microbiome. And this is at the bottom here. You can sort of see the changes in the microbiome. So this is what it would look like in a normal non-treated mice. You see the changes that are coming from giving them this agent, and this is the microbiome in the fasting mimicking diet DSS mice, and you can see an increase in this lactobacillus population here, and you can also see a population of bacteria that is actually only found in these FMD treated animals, and this is the bifidobacteria here. So when you actually look into some of the research that's published, both of these bifidobacteria as well as the lactobacillus, both of them, an increase in these populations has been associated with reduced incidence of inflammatory bowel disease. And we followed this up by actually just doing an oral gavage into the mouse with these fecal transplants. And we could show that only animals that received the fasting mimicking diet prior to the oral gavage, and then were exposed again to this drug here. In this case, these animals actually again had lower incidences of inflammatory bowel disease, and again we see all these phenomena occur. So the next few slides, I call them hot off the press because these are publications that are just out for about 2 to 3 months now. So this is sort of the brand new work. All of these are currently focused on cancer, and we have done those in collaboration with another lab in Italy. So the first one that I want to show you, I think is really interesting, and this is talking about using vitamin C treatment and the fasting mimicking diet against KRAS mutated cancers. So vitamin C has sort of emerged lately as a efficient drug, or I mean, whether you want to call it a drug or not, but it's efficient in reducing tumor progression, in particular for these KRAS mutated cancers. And it's important to note that this effect of vitamin C is because it has a pro-oxidant action, so it's inducing reactive oxidant species. And this is largely or strictly dependent on metal iron redox chemistry. So I'm not going to go into full detail what's happening here. I'm just going to show you that using the FMD together with vitamin C in these colorectal cancer cell lines has synergistic effects. So you can see here, this is sort of the same style of graphing that I showed you before for the breast and melanoma cancer. Black here are mice that are just fed without any limitations. In orange, these animals have been given vitamin C. The red line, which overlaps with the vitamin C treatment, is just the fasting mimicking diet. And again, combining the fasting mimicking diet together with vitamin C has synergistic effects and dramatically reduces the tumor volume. So this goes in hand with an increase in reactive oxygen species, and this is particularly coming from this is in vitro, so in cell culture. This is coming from we use short-term starvation in vitro to mimic the effects of the fasting mimicking diet. And you can see that this starvation in these cell lines induces reactive oxygen species, particularly when you combine the two approaches, so fasting together with vitamin C. And the important thing here is that this increase in reactive oxygen species goes in hand with a reduction in the heavy chain of ferritin, which plays a role in this metal iron redox chemistry. So the relevance here is, without taking this too far, that low levels of that the fasting mimicking diet with the vitamin C induces low levels of this ferritin heavy chain. And this is important. So these are it's a comparison between the KRAS wild type and the KRAS mutant patient. And you can see that it doesn't really matter in the wild type whether or not you have high or low expression levels of the ferritin heavy chain. However, in patients that have the mutated form of KRAS, you can see that 3-year overall survival is actually longer for those that have low FDH levels. So, meaning that the outcomes of the combination with of the fasting mimicking diet together with vitamin C likely also has a better prognosis for patients that have this. So, meaning the treatment efficacy is improved. More importantly, this is a little bit of a hectic graph, but you can ignore the top 4 because it's sort of what I showed you here already. But when we now combine one of the drugs that is commonly used for the treatment of colorectal cancer with OXP here, oxaliplatin, when you combine all 3, vitamin C, FMD, and OXP, you see this nice overall reduction in cancer progression over time. So again, we can use multiple approaches to target cancer progression. This one is the last publication that we have in Nature, together with Alessio Nencioni's group in Italy, and this is essentially using the fasting mimicking diet in a breast cancer setting combined with endocrine therapy. So the problem is that while about 75% or so of all breast cancers are hormone receptor positive, meaning they express these hormone receptors, they over time can require endocrine resistance. So this is largely because these pro-growth pathways that I showed you before may lead to the dysregulation of cell cycle regulators, or you can also acquire mutations such as, for example, in the estrogen receptor encoding gene, which then leads to a failure of these endocrine therapies because they're simply no longer responsive. And this is true even when you add cell cycle regulators or for example, TOR inhibitors. So this over time leads to tumor progression. So the problem here is that acquired endocrine resistance is quite common, and it's a major problem. So what we did here, I think, is really a really interesting study. So you can see here, MCF7 is a breast cancer cell line that is hormone receptor positive. This is just a control, and you can sort of see it just grows over time. It's doing what it's supposed to be doing. And for the first sort of 65, 70 days, the combination of fulvestrant together with palbociclib, I always miss it up. This we call PALP, sorry. So the tumor is well maintained and is essentially not growing. But then after that, we see this burst in growth, right? So at this point here, the tumor has essentially become resistant to the treatment, and it's growing quite fast. So what we did at this point was a biopsy. So we took some cells out, expanded them in cell culture, and then simply combined the fasting mimicking diet with these two drugs. And you can see at this point, when we then put the FMD back on it, now we're beginning again to reduce tumor progression and the tumor shrinks. When you take these cells here that were isolated from the biopsies, put them back into the mouse and let them grow, the green line here, even though you give them again the drug or both drugs, you can see these are resistant. They're growing, right? They're growing nearly at the same rate as these untreated cells would be about the same period of time. So about 25, 30 days, you can see the growth rate is somewhat comparable. But after 10 days of growing these cells in the mouse, now we did 3 cycles again of the fasting mimicking diet, and here now the tumor again becomes responsive to the treatment. So meaning the fasting mimicking diet in this setting, when combined with endocrine therapy, restores sensitivity in resistant tumors. And this is just the results from one of the patients that underwent it. So just call him patient number 26. So he had second line treatment for hormone receptor positive, HER2 negative breast cancer, treated with fulvestrant, PALP, and FMD. This is on top here. You can see the baseline. You can see sort of the cancers, and then the patient underwent 8 FMD cycles. So by month 11, is essentially a clinical control disease. And there's some other reports that this happened. Again, there's a need for a large clinical trial to verify all this, but these are some of the results that were published. And the sort of taking this further, this is the direct trial. So this was done in the Netherlands. This is a FMD as an adjunct to chemotherapy for breast cancer. This is sort of the consort. So you can see about 131 subjects. The primary endpoint of the study was focused on toxicity, so grade 3 and 4. And the conclusion was that actually the untreated or normal diet and FMD had comparable grade 3 and 4 toxicity between the groups. But the important thing here is that the FMD cohort actually did not receive dexamethasone. So dexamethasone is often prescribed to sort of reduce the side effects stemming from chemotherapy. So in this case here, despite the fact that that the primary endpoints was comparable, the FMD group actually did not receive dexamethasone. So you could have, it might mask some of the effects that the normally eating subjects received the drug. The really important thing here is looking into sort of the tumor response data. And ITT is the intention to treat. and PP here is per protocol. So the Miller and Payne readout here is essentially measuring how much of the tumor was responsive. And when you compare the sorry, MP4 and 5 is the higher response than 1, 2, and 3. So you can see here in the intention to treat, we have a little bit higher number of patients that are in the MP4 and 5. So this is what we want to see. But when you actually split the data up and look at the per protocol, meaning were they actually compliant with the FMD or not compliant compared to the regular diet, you can see that those subjects that were high in the compliance with the fasting mimicking diet had a higher proportion of patients that were actually responding to their treatment, and so that's what you want to see. This is good. And then when looking at the radiological response, we sort of see the same effect. Again, those people that were compliant with the diet had a higher complete or partial response to, we had less of these sort of solid disease or progressing disease states. So yeah, you can see the proportion here. So FMD, those that were compliant, higher radiological response. Those that were not compliant still underwent 2 to 3 cycles, so they're having some benefit from it, but clearly not as much as adhering to the diet. And then these are the subjects in the regular diet. So should have done that. Sorry. So yeah, this is highlighting that the FMD in this setting really improved the tumor response in this trial. Okay, I'll sort of go through this. This was the first trial that we published on just using the fasting mimicking diet in about 100 volunteers, and we really just wanted to see whether or not this affects markers associated with healthy aging. The idea was here we did 5 days of the fasting mimicking diet followed by 25 days of normal unrestricted food intake. We didn't give them any suggestions on what to eat, and this was a randomized crossover trial. At baseline, the subjects were comparable with regards to the distribution of males and females, comparable age, body weight, education, and so on. When looking at the dropouts, there was no difference in sort of dropouts versus completed. In gray, you can see these are the people that withdrew or dropped out. There was really no difference in age, the BMI, but a higher percentage of females dropped out, and we're not quite sure why that is. Just to really highlight that even in humans, the FMD is doing what it's supposed to be doing. We see a reduction in glucose, an increase in ketone bodies, a reduction in IGF-1, and an increase in IGF-BP1. So again, these are the same markers that we looked at in mice. Mice have a lot higher metabolism, so the changes that we see in humans are not quite as dramatic, but they're still going in the same directions. And so these are the effects that you or that we saw after 3 cycles on the fasting mimicking diet. So obviously it's a low-calorie diet, so we see a decrease in body weight, a decrease in the body mass index, see a reduction in total body fat, and particularly trunk fat. There was no significant change in the lean body mass. See a reduction in waist circumference. Lean body mass is important. A lot of the diets, obviously, people often lose lean body mass. We did not see that. So yeah, reduction in waist circumference. Glucose here, there's no change, but it has to be said that after 3 cycles, when we measured, these people were already back on their normal diet for 5 to 7 days. So you really wouldn't expect them to have still reduced levels in glucose. Insulin-like growth factor 1, we see a reduction here. Changes in systolic and diastolic blood pressure. No real changes in sort of the lipids, triglycerides, total cholesterol, LDL, HDL. No real changes. CRP is a marker for sort of systemic inflammation, C-reactive protein. After the 3 cycles here, we didn't see significant effects, but we went back and we did a post-hoc analysis where we stratified subjects by risk groups, and so you can really see here that the FMD seems to be particularly efficient in those subjects that actually need sort of a modulation, right? So all of these subjects here that you see underwent the FMD, and then you just split them up. So you can see here BMI, normal weight, overweight, obese. Naturally, obese people obese subjects lose more BMI. Those people that have high systolic blood pressure over 120 have a higher reduction than those that were sort of in the normal range. Diastolic blood pressure the same. There is no real clinical cutoff for IGF-1, but there are multiple studies that show that values over 220, 225 are associated with a higher cancer incidence. In this case here, we see it again in those subjects that were over 225 nanogram per ml, reduction in IGF-1 levels. Glucose here, this is actually, I thought this was one of the cooler things. For those that were considered to be pre-diabetic, so over 99 milligram per deciliter glucose, in this case, they had quite a dramatic decrease in blood glucose levels. Triglycerides, if you split it up, yes, over 100 goes up. Total cholesterol, those that are over 199, significant reduction. LDL levels decrease, HDL levels. And again here, CRP now, when we split it up between a low risk to moderate and high risk group, those people that were over 1 for CRP levels, we see a significant reduction in CRP levels, so indicating that we're reducing systemic inflammation. So just as a real conclusion, and I have 3 more slides after this, and I'm done. I think I'm still good in time. So I think I hope that I could show you that diets are a powerful tool to prevent and delay many of the aging-related diseases. I hope I showed you that the fasting mimicking diet improved the efficacy of cancer treatment. It reduced intestinal inflammation, increased regenerative markers in the IBD, inflammatory bowel disease mouse model, reduces symptoms in the multiple sclerosis mouse model, and it's reducing risk factors for diabetes, cancer, cardiovascular disease in clinical studies. And so at the bottom here, you can sort of see the preclinical findings summarized, right? So I've shown you that type 1, type 2 diabetes reversed, multiple sclerosis, chemotoxicity, and sensitization to chemotherapy. So naturally, the question is, where do we go with this? So one of the things that this is published, this is essentially a proposal for the use of the fasting mimicking diet in diabetes. This trial has a trial identifier, so this is hopefully enrolling soon. And you can see here that sort of the treatment, essentially a fasting mimicking diet followed by the regular diet, 12 cycles, so about a year, compared to the control group, with the primary outcome focused on HbA1c and type 2 diabetes medication dosage, and then a bunch of secondary outcomes. So this trial is hopefully running soon, and this is also going to be in the Netherlands. We just finished up a trial together with Dr. Mark Houston at the Hypertension Institute. In this case, the idea was to test whether periodic fasting mimicking diets would be as beneficial as a Mediterranean diet in subjects at risk for cardiovascular disease. The reason here is that Mediterranean diets often sort of are identified as the most heart healthy diets, and we just compared this. The primary outcomes were all focused on endothelial function and material compliance, and then secondary outcomes, changes in cardiometabolic risk factors. And this is sort of unpublished and preliminary, but it's based on the initial analysis that we did. And so we included 84 overweight obese adults. The enrollment criteria: poor endothelial function and low arterial elasticity. And both the fasting mimicking diet and the Mediterranean diet decreased body weight and fat and improved arterial elasticity, and tended to decrease the reactive hyperemia index. The Mediterranean diet was more effective on blood pressure, but reduced lean mass. The fasting mimicking diet seemed to be more efficient on cardiometabolic risk factors and preserved muscle mass. So This is not yet published, but we're submitting this probably in the next week or so. So things might still change, so take it with a grain of salt. But this is sort of the outcomes. This is something that I'm quite excited about because I've been working with a colleague to sort of test whether the fasting mimicking diet reduces biological age. So when I mean biological age, it's essentially the idea that chronological age, meaning when you were born, so your years in age, is not a good proxy for so your actual organismal age, right? So it's like on the cartoon here, you can see that there's a lot of variability. They're probably all within the same sort of age group, but there can be quite dramatic changes. So the idea is to use biological age, and that this aligns with essentially a measure for aging for basic research and screens. And you can facilitate the evaluation of interventions aimed at delaying aging. So you can measure, can you actively reduce biological age? And it's also a basic aging mechanism. So the results that we have by simply using some of the biomarkers is that that a projection, which is it's essentially mathematical modeling here, that completion of 3 FMD cycles per year between the age 50 and age 70 increase your lifespan by about 5 years when you get to age 70. So just by modeling repeated cycles of the FMD and the changes that we see in the biomarkers that come with this, we're expecting that doing this for 20 years makes you gain 5 years when you get to 70. Here's an overview of some of the ongoing clinical trials, largely focused on cancer right now. A bunch of them are in Italy. Some of them are here at USC, and some of them in the Netherlands and Ireland. So most of them are still ongoing, and we're waiting for results. That is really all I have. These are acknowledgements. The Fasting Mimicking Diet is provided by Nutra to us. We don't pay for it, but we're doing the research. I'm supported by the American Federation for Aging. These are sponsors for the lab, and that is all I have.
And that was amazing, Sebastian. See,
thank you.
What is so great? Sorry?
I'm sorry if I rushed through it.
Oh, we loved it. You have kept about 170 people in their seat for all the time you've been talking. So thank you very much. That's excellent. And of course, lots of activity have been going on the side and the questions. And Inge, my good friend in Norway, he has been persistent in a question about ketones. And he's asking why the FMD diet was not designed more as a ketogenic diet, and if the FMD pushed the ketones even higher than what it does, would we get even more benefits? So we come into this question like, why have you chosen the diet the way you have, kind of?
Yeah. Okay. So yes, we do think that the initial sort of the one reason why particularly the day one of the FMD is a little bit higher in calories, and a lot of the calories are coming from fat, was because we wanted to help the body get into it's not really ketosis state, but it's just have higher levels of ketones around, because these can be used as energy. So the idea was to reduce the sort of slacking and feeling less energetic in humans. It's one component to this. The reason why the diet isn't a full ketogenic diet is simply because we don't think that we have any indication from longitudinal studies and epidemiological data that long-lived populations of humans actually undergo ketogenic diets. So part of the reason why I said that we want to have the research, or that we want to have the diet based on healthy, or knowing what healthy aging looks like, in particular coming from some of the blue zones. A lot of these are simply not undergoing ketogenic diets, and there's not a lot of data that supports that ketogenic diet actually induce longevity. I don't think there's a lot of data from animal studies, and there's not a lot of data in humans. So while I think it's good for sort of short-term weight loss in humans, it's like, yes, but we don't know whether or not these effects actually translate to an extension in longevity. And that's the main reason why we're not designing a ketogenic diet. The idea is still that a balanced diet is important in our opinion, and the other concept obviously was that initially the idea for the FMD was that it was more like a anti-cancer approach, and so the protein was our main focus. We wanted that to be low, and carbs were supposed to be low, so naturally it has to come from fats somehow, but it's not. Yeah, we just didn't want it to be fully ketogenic. I hope that answers it. It's a little, yeah, it's a little tricky, right? I mean, we could go higher in the fats, but then the problem is when you look at what people eat for ketogenic diets nowadays, it can be very confusing too. It's like, I mean, I'm not going to go out and tell people you should be eating a lot of bacon because it's high in fat, knowing that processed red meats are probably the worst thing you could put in your body.
Excellent. So a few questions that I'm going to try and bundle it, because I've had several questions like, what if one has hypothyroid, and what if you're stressed, and so all these like suboptimal health conditions in regards to, and then using the Prolon, which is effectively is a stress factor, but it becomes one of these like healthy stress factors because you're not doing it that long. So if someone has hypothyroidism and they are balanced and on with their T4 medication, would you then advise them to use the FMD, or is it one of these where you're saying, well, the research hasn't been done?
Yeah, I mean, so I would put two things out to this. One, I'm not a medical doctor. I'm not a treating physician, so I don't know the case, and I'm not gonna go out and say yes, do it. I think there's a lot of strong data supporting that the FMD is safe, but I think that's a decision that an actual physician has to make. And you know we're always very supportive, and we're trying to provide data, but I don't think that we have anything right now on hypothyroidism. It's almost too specific. There might be cases of people that have taken FMD and have no side effects, or might have some. I usually don't see them. I think that's a question that could be directed towards El Nutra without name dropping, but they usually follow up with reports that people don't handle Prolon well. So that's not endorsing them, just putting it out there. But I think there's you could follow that up.
So the challenge here is that we are sitting as practitioners with many different health conditions, and the research just isn't there. So very often we have to evaluate, is this a good thing for this person or not? Is the person very, very stressed out and the adrenals are hanging in a thin thread? And would this stress doing fasting for 5 days, would that add even more stress to a stressful body already? Yet again, if you have been diagnosed with breast cancer, you're pretty stressed already because you've just been given a disease, and that seemed to work well, at least for the condition and the treatment of the cancer. So it's this really balance, like where, and that is always like that in a way with diet. I feel that, oh, there's some research that indicates, but there isn't enough because we need money to do the research in nutrition, and we can't trademark it and like if it's a drug and so on. So we always end up with a lot of questions where when we're treating with a dietary strategy, and I guess that's what you're also demonstrating here. You're demonstrating exactly how far are we in the research, and then of course we are a little bit left on the side as practitioners as when should we treat and when not.
I mean, again, I think some of the data that we have, even though it's preclinical, the idea obviously is that fasting and fasting mimicking diets increase resilience, right, and stress, and there could be a component to it which is almost like a hormesis, right? You induce a mild stress to be able to deal with higher levels of stress. And yeah, it's a shock to the sort of the environment with both approaches. But again, we're not reinventing the wheel. Like I think fasting is around. Our body knows how to cope with it, and unlike some of the other dietary approaches, it is a short period of time. So I think without saying that's what you should do, I think there's a way if the physician feels like I can supervise it for 4 days, 5 days, get a report and see how the patient is responding, then that might be sort of directing where to go with this. The problem for, yeah, like you said, the problem for us is always going to be the need for us to do solid sort of preclinical research, which often ignores the fact that we can't. It's not a very specialized or individualized approach, right? I mean, it's the same with we're using cancer models and we're using standard drugs, but there's also humans receive chemotherapy differently than a mouse would. It's different regimens. So often for us, it's an issue if people say, "Hey, I want to do this, but my treatment plan is 8 days." I'm like, I can't have you fast for 8 days without anyone looking at you. So that's always the limiting part. We have a lot of freedom by doing preclinical research, and every time we do a clinical trial, we see how complicated it gets to actually just translate it, even with a dietary approach that was designed to be easily translatable.
So a last question is more general in regards to like autoimmune conditions, because some of the, and I guess we're still lacking the research to really answer this question, because some of the theories is that so it's like pre-cancer cells that are removed when we go into the fasting state, and autoimmune cells as well. And is that still what you are thinking, even though in autoimmune conditions there's so many, and like in cancer there's so many different types, but is that the take home message as well in regards to autoimmunity?
Yeah, I think with regards to autoimmunity, we've seen it on multiple levels, that it's not just targeting one component of the diet, or one component of the disease. Sorry, it has, just because it has broad effects, we usually see it sort of target multiple things. Like the multiple sclerosis study that I mentioned had a clinical arm to it, where the patients underwent 7 days of a fasting mimicking diet and followed by a Mediterranean diet, and they self-reported a dramatic improvement in their quality of life compared to sort of a ketogenic diet. And yeah, I think that the answer is that I think it is efficient. One sort of idea that I've been working on a little bit now is, given that in hand with the fasting mimicking diet, we see sort of a loss of cell mass, and I've been always intrigued by sort of, is it a survival of the fittest, right? So a normal cell can always really easily adapt to a fasting environment, but particularly when we look at cancer cells, they're so specialized and develop in a environmental niche that actually allows them to continuously grow, just because there's a consistent supply of nutrients around. And they actually cancer cells do really horrible when you restrict them. So there seems to be sort of the idea that the accumulation of mutations, while in a nutrient-rich environment, is a good thing. It is detrimental to them in a fasted environment. So the same sort of, like you mentioned, like pre-cancerous cells, so we'll still have aging deficits or mutations. I think there's a great window there to target these with fasting and eliminate these cells. And then when you refeed mice, we see regeneration, and that's hopefully then stemming from these younger cells that can respond better to these changes in the environment.
Survival of the fittest. I like that you bring that up. I also want to remember this hormetic stress, the healthy stress. Heat is hormetic stress, fasting is hormetic stress, ice baths are hormetic stress, exercise is hormetic stress, and so on. So we have to look at fasting as being a hormetic stressor, and it's gonna make us stronger in a way. So there's more questions, but I think it's time, Sebastian, for you to go on 4 days holiday. Sebastian has, this is really from his heart, so because it's his day off really, so he should run off now and enjoy, and we're grateful and thankful for you to spend this time with us. And yeah, we are looking forward on the side to see all the research that will be continued published, and maybe in a few years time I'll invite you back to get a follow-up. So thank you very much, Sebastian. Thank you very much, everybody out there. We'll do the virtual hug. Take care, and we'll see each other hopefully in 2 weeks time again. And look after yourself and everything. Bye.
Right. Thank you so much for having me.